questions? No, sorry.
Yes, okay.
Fascinating material, thank you. And I have a number of thoughts, and some of them are questions. First, going back to the preferred versus non-preferred, and resulting in different reproductive output, it seems to me that you need a control condition there which is solitary, because it could be the case that having a non-preferred partner is stressful. And so it's not that the preferred partners are helpful, but that the non-preferred partners are simply suffering, inflicting a cost. Without that, it's hard to interpret what that is.
Right, yes. I mean, I see what you mean. I mean, we just relied here more on the fact that we took our lifetime experiment, lifetime reproductive success experiments from what I presented at the beginning. And it's actually the case that they are not significantly lower than the monogamous females. I mean, of course, that's a different, it's done at a different time, but
serves as a control.
Right.
Okay.
But because you're mentioning that point, and that's why I presented that slide here, the interesting thing is that actually if you're a preferred partner, it doesn't matter. I mean, you always can, you have two females, and one is always better than the other. So I call them just here superior and inferior, just by looking at the numbers that they produce. And the interesting thing is that actually if you're together with a preferred partner, it doesn't matter whether you're first or second or whatever, you in the end, on average, have a very similar success. But with a non-preferred partner, one is actually suffering. So on average, you're on the level of a monogamous one. What one is actually worse. And this may indicate that we might observe under specific situation that they might try to leave. Yeah.
Okay.
But on average, it's like a monogamous. Yeah.
Well, you handled that one so smoothly. Let me try again.
Good.
So on the B cell T cell difference with your, they're not heterozygous, but we can think of them as
Yes.
socially heterozygous. It seems to me that while it's an interesting result, it can't be an ultimate explanation of the alloparenting, because if there's some advantage to having one particular combination of B-T cell ratios, then selection should just favor that over time, right? That is, if you do better if you're at the intermediate level, which is your socially heterozygous individual, then over time, selection should simply favor that level of B cell and T cell. So I don't, it's an interesting result.
Okay. I get that point.
But I don't think that it can provide an ultimate explanation. I'm more sympathetic to the remark that you made in passing, and I think Wayne Potts would probably endorse that, namely that there probably are functional consequences to differences in immunocompetence as a result of social experience, but that you don't see them in the laboratory because they're not exposed to a sufficient number of pathogens and so on. So if that's the case, supposing that that in fact is driving this behavior, there then is, it seems to me, an analogy to the optimal outbreeding problem, because the last study that you showed us tells us that they're using propinquity during nursing as a cue of some kind of either relatedness or social attractiveness, right? Because they aren't distinguishing on the basis of MHC. That means that if we think about it as kin recognition gone awry, right? So presumably in the ancestral trait where they're all monogamous, then your litter mates, just the experience of nursing together is a good cue of relatedness, and then you have a derived trait of alloparenting, and that ancestral trait hasn't been sufficiently modified, so they don't distinguish among litter mates. Right? Well, now you have a benefit if the immunocompetence hypothesis is right. You have the benefit of co-parenting, in that your offspring have a wider array of maternally derived antibodies, right? But you have the cost that your offspring will misidentify as kin individuals to whom they're not related, right? So there's going to be an optimal degree of relatedness, just like there is in the case of inbreeding, outbreeding, right? Where the benefits of MHC heterozygosity, social heterozygosity, are matched with the costs of identifying as kin individuals to whom you are less than fully related.
I think a lot of points are certainly signed. First of all, I think with the experiment of the immunocompetence hypothesis, we are not thinking the intermediate level is the better one. I mean, it's just showing there is an effect, which is the start of it. And the basic idea, to coming back to your second point, is probably what they do is to have some complementary decision, whatever. So it depends on your own MHC, what you choose in another partner. And it's obviously not the case that they always go for the more similar one. So maybe among sisters, they choose the one that best complements your own MHC. And this might also be influenced by the environment, whether parasites are around or not. And so this would actually explain why they're not just going for the more similar one, like in our full sisters and an unrelated female. We didn't check in this experiment for the MHC. We are now looking in a wild population actually for MHC patterns in animals that can roam freely. But I can't tell you the result yet. So, I mean, here we didn't actually analyze the MHC of these animals. So, unfortunately, in that experiment, you know, we just manipulated. We just have had these two sisters and one unrelated female. But the idea here is that they might do have some kind of information about their own MHC and choose among those with whom they grow up somebody who is complementary, whatever that is, but obviously not totally different from yourself. I mean, in mate choice, now there's accumulating information that, like in sticklebacks and also in house mice, that they actually do that they use some complementary setup of MHC. So you can't predict from one, it's not that it's always the same individual that's attractive for everybody, and that's what we see in the preference test. And this can end up in the end, as you said, in some kind of these optimal degree, or whether it's optimal, it should be, if immunocompetence plays a role, it should be somehow optimal. But it doesn't really say it's the medium level. I think it's just here quite nice to see it does influence the immunocompetence of the pups. And that's actually what our first, what we were pretty excited about, that it's worthwhile looking at this in more detail. And we actually now try to get more information on that. And this also explains why, first of all, they're not all going for the same female. Second of all, it's not that they're going for the most diverse MHC type or the more similar one. So there seems to be something going on maybe on this kind of complementary.
In wild conditions, is there multiple paternity within a single litter? Yes. So you can have litter mates who are divergent.
Yes, we get actually, that's not yet published, we get quite high multiple paternity of about almost 40% of the litters have more than one father. We could analyze up to now up to 4 different fathers per litter, which is quite, I think, quite amazing, because people have thought up to now that females are in estrus for a very short period of time, which would not allow to mate maybe for more than 1 or maximally 2 mates, but obviously they do. And the other point is that they do not always communally nurse, even if they have the option available. So there seem to be costs involved, and that's the other point. And these costs might also be that you transfer parasites if you allow, everybody into your nest. So there seems to be some kind of a trade-off. If the ideal partner is not available, you rather probably nurse solitarily. So it's not as simple as we thought from the lab, if you then go to a field population. Yes?
Have you considered age effects? I mean, am I correct in thinking that what you've done here were all same age?
Yes.
And is that the way it is in the wild? And might there be in a communal nest a preference by a newly reproducing female for an older female?
Mm-hmm. What we did is, in the lab, is we also looked at mother-daughter pairs. Of course, as you said, not just as sister pairs. And if you have a mother and daughter, you get a higher potential for more despotic societies. It's to say it rather simple, young females avoid, if they can, to communally nurse with an older female. And I mean, under natural conditions, we did a lot of experiments now even with sisters, full sisters from different litters, but genetical full sisters that differ, that never met before. And they try to avoid that, they don't like that. And I thought quite a lot about it. Why don't they like an unfamiliar sister? I mean, from the behavior, you get indication that they can tell the difference between an unfamiliar sister and an unfamiliar, unrelated individual. But the point is that the litter size of the second and up to fifth litter is larger than the first litter size. So as a maturing female, if I meet an unfamiliar sister, she, with a very high probability, will be older than I am, and she probably has had a litter already. Now, during communal nursing, they're sharing the burden of lactation equally. I will have a smaller litter, the other one will have a larger litter. This means I will over-invest into the first communal nest. This might explain why they actually prefer to go for somebody with whom they grew up instead of staying with the mother. In our wild population, we very rarely observe actually mother-daughter pairs to communally nurse. It seems to be something, because then you're over-investing for your first litter at least. But we haven't tested that hypothesis yet. But it's the only explanation I have why they don't like.
The younger mother is actually putting out the same volume and quality of milk.
Right. I mean, to make it simple, if you have 4 pups and the other one has 8, both will produce milk for 6. Yeah. So if you have the smaller litter, you overinvest. I mean, that's the other idea. We predict that they actually choose also partners according to litter size. But they should be able to have some kind of an idea before they give birth on litter size. And that's something maybe you have more experience with, or some of you in working with primates, what kind of signals? I mean, of course, you can have different hormones, oxytocin, prolactin, that might signal during pregnancy something about, potential lactation abilities or litter size or so. We have no idea yet whether they can tell can have information about the litter size of another female. Yep.
Is litter size related to body size in the females?
No, not in the wild ones. I mean, in some of these inbred strains you get these effects, but in the wild ones not. At least there's no clear effect, but there are very clear fertility differences between females. And what we learned is from the experiment on the peak load reduction is if you standardize litter size, you don't standardize investment. I mean, that's the main message in the wild females. You have rather small females who can produce 2 or 3 litters of 8, 9 pups consecutively without any problems. You have others that produce a litter of 7, then they go down to 4, and then maybe to 6 or something. There are huge fertility differences also in our natural population. And of course, I mean, there is some kind of rough correlation. If you look at lactation, then those with a larger litter have a higher body size. But you can't really predict it at maturity.
So is there a terminal investment increase, and does it take the form of lactation? Because if so, then there's an endgame strategy if you're a mature female, which is to try and choose a partner who's on her last litter.
Right.
Because you get a boost from her increased investment, and you probably won't have to pay it back because she's at the end. Right?
We have no indication that there is senescence or something like terminal, that they know when they give birth to their last litter. So we looked at old females and we looked at younger females. The only thing you get, and that in the lab you get senescence in the sense that you get a decrease in litter size. We do not know yet, because we haven't finished all genetic analysis of our wild population, whether they live long enough to really see this kind of senescence going on. But that's of course a very interesting idea to look at. I mean, what's actually a good partner? I mean, somebody who just says, okay, I can invest everything I still have. Yeah.
And by the same token, in the wild, do we know that their social relationships are temporally stable, sufficient that there's likely reciprocity over time?
Yeah, I mean, we know it from our population that you get reciprocity. And I mean, it's just from it hasn't been published before. There's no information on other populations yet. But there is the potential, and social groups are pretty stable as long as the animals live. We do have some females who live now for over 2 years. in a barn. It's an open barn system. They can, and inside there is no predation. Outside, of course, there is, but I think that's pretty normal for house mice. And most of them die pretty young, but some of them manage to live for over 2 years, and they live in pretty stable groups. Yeah. And so there is the potential for reciprocity.
I was just curious, were there any differential costs to females feeding drill and us? Does it matter at all, or does it matter?
Yeah, you mean, I mean, the only thing I can say is that they have to find the food every day they invest into lactation. They don't store food, they almost have no fat storage or something like this. So they have to search for food every day and get all the amount they need to invest into their milk. And of course, litter size has an effect on the amount of food you need. And what you're aiming at is how probably confident we can be that they equally share the duties of lactation. And I'm not quite sure whether I have that slide here, probably not. We did one experiment where we, yeah, okay, I have it here, where we looked at the food consumption of females during communal nursing. We have set up a device where we can measure individual food consumption of females, even when they lived in social groups. I'm not going into details about that at the moment. But if you look at the food consumption of the female with a younger litter in a communal nest, you see that the red dots here is the food consumption as a function of the production of litter mass, let's say the total body mass of pups produced. And the red dots refer to the own litter, the litter of our focus female, and the blue dots refer to the situation of both litters together, the entire communal nest. And we only get a significant correlation between food consumption and production of litter mass if you take both litters together. So this signals that it's not own litter size that regulates the investment of the female, but it's the size of the entire communal nest divided by 2. That's the actually best evidence at the moment we have to really say that they're equally sharing the investment. And this means one female, by definition, is always over-investing and the other one saves. And I mean, this of course comes back to the point, if it's reciprocal, it's okay. And if we consider that, okay, even if you're the inferior one in a pair of 2 egalitarian females, you're better off than a monogamous female. I mean, even if you would be even better off if you're the superior one. But even in the situation of being the inferior one and over-investing in the communal nest, you have an advantage in comparison to the solitary situation.
Right. Yeah.
So they do differ, of course, in their individual food consumption, but I think in terms of where selection works on average, you're better off than in a monogamous situation.
Okay.
Yeah?
I was wondering if the social bonds between the nursing partners have already existed before they get communal nursing, or they just randomly pair it together after that?
Yeah. If you would have invited me 1 year ago, I would have said yes. But I know now, no, it's not the case. And they even can change partners. They do seem to establish, I mean, the picture is they live in these social groups. And they socially interact with all group members. They do rarely interact with non-group members, but they establish social bonds within their social group. But only when they're pregnant, then we start to see these interactions that can result in communal nursing. And that's really interesting. I mean, I thought that they might establish these bonds when they mature and then they stay with that partner. And okay, if the partner is not giving birth, then you don't have the option for communal nursing. But they can change partners actually, and even if there is an option for communal nursing, they don't always take it. So they even nurse solitarily, even if another female is there. And if we look only at the social bonds, I mean, what we do is actually doing social network analysis now to just see who has a stronger bond in terms of spending more time together in a nest box in that free living population. And here pregnancy seems to be the critical phase. But what's actually going on there, I have no idea. But the only thing I can say is that they change partners, social partners. So you don't always stay with your previous one. And that's of course exciting. I mean, why do you divorce from your social partner? Maybe you have had the smaller litter, and then, but because we don't have all maternity data yet analyzed, I can't tell you.
But you have to be, in choosing a partner, you have to be out of phase with them, right? So
Yes.
otherwise, the peak load benefits
Right,
won't
right.
happen.
Yeah, but I mean, I thought about that, and then I can't really come up with some kind of evidence whether these 8 days are selected or not, because you always get an average value. You know, if you just take, I mean, what we observe is that communal nests very rarely of litters of the same age. That's what we observe. And being out of phase, it seems not to be too difficult. I mean, we get these group sizes of having up to 8 females or something like this in the same group. They do have seasonal effects. I mean, in winter they actually go down with reproduction. In spring, but yeah, then you need quite a large sample size. We only have 3 years now where we could, and we actually intended also to look whether there is some kind of synchrony factor, so but statistically it's almost impossible yet to analyze that. But it seems not to be very, not to really be a problem. I mean, they're certainly not triggered in terms of that they all go into estrus on the same day or something like that. But yeah, whether there's selection on that, I can't tell you. Yeah. Yeah.
Are there any increases or decreases in infanticide of the pups in the 1 versus 2 female families?
In the free living population, I can't tell you yet. Because sometimes you have 2 females visiting a box and we do not, I mean, we are analyzing at the moment maternity, so we don't know yet. And the consequences for infanticide, my gut feeling at the moment is, I mean, Jo Manning published, she provided data saying that in the communal situation they are better protected against infanticide. What we observe is even infanticide among the cooperating females. And my gut feeling is that in our population we don't get that effect. I mean, what's happening actually is if you have 1 female gives birth, because they are non-synchronized, whatever it is, and the other is still pregnant. And what we observe both in the lab and also in the field is that the highly pregnant females, shortly before they give birth, sometimes kill 1 or 2 of the already present pups. And it's quite a striking behavior because they, before that, or go there, they huddle over the pups, maybe they give colostrum, we don't know, they lick the pups, and then there seems to be a rather small time window before they give birth, when they suddenly, I mean, they usually grab a pup, carry it to another place, lick it until licking gets very heavily, and then they kill the pup by a neck bite. They don't eat these pups. It doesn't look as if they are really looking for proteins or so. They just drop them, and you get these, I mean, you then find these poor dead pups, you know, being killed by a neck bite. And then they go back after 1 or 2 hours and lick the other ones and give birth, and then everything is very nice and very friendly. And this illustrates that they're obviously not able to tell apart own from non-offspring, because after both have given birth, we don't observe this kind of infanticide anymore. So they do seem to compete over the number of own pups in the communal nest.
Okay, we're just about out of time, so
Sure.
thank you very much.